We introduce AnimalClue, recognizing animals by their traces, presented at ICCV 2025. AnimalClue includes footprints, feces, eggs, bones, and feathers, totaling 159,605 bounding boxes from 968 species and annotations for 22 traits. Identifying diverse animal species remains challenging, as most models overlook nocturnal or camouflaged animals. Wildlife monitoring can rely on indirect evidence like footprints or feathers, but this requires highly domain-specific knowledge. Since manual inspection is not scalable, there is a strong need for vision models that can identify species from such traces. Recent advances in computer vision have greatly contributed to automating key wildlife observation tasks. However, identifying species from indirect evidence, like footprints or feces, remains relatively underexplored, even though it plays an important role in wildlife monitoring. Several previous studies have tackled trace recognition. However, their species coverage and bounding boxes are limited, and most datasets focus only on classification, which is not sufficient for realistic wildlife monitoring scenarios. In contrast, our AnimalClue dataset covers 5 major trace types, provides segmentation masks, and 22 biological traits of species. Firstly, we collected images and their species labels from iNaturalist, selecting only those that meet both conditions, research grade and Creative Commons license. After that, we used grounding Dino and segment anything to create initial masks. However, for footprints, grounding Dino often failed to detect them correctly, so manual annotation was also performed for the initial masks. After creating the initial masks, we manually corrected the mistaken ones. During this process, we also removed irrelevant images, such as those containing overlaid text, animals in the frame, distant objects, or visible human faces. This is an example of the final dataset. Our dataset consists of 968 species, 200 families, and 65 orders. It includes a total of 159,605 bounding boxes across 5 trace types. This shows the top 50 species distribution in our AnimalClue dataset, with the vertical axis representing the log frequency. Let's move on to the experiment part. This is the classification result. Swenby achieves the highest top 1 accuracy among all baselines. Feathers show the best accuracy across taxonomic levels, followed by eggs, which also have the second largest number of species. This is a visualization of TSNE. As you can see, our fine-tuned clip model achieves the best feature separation across categories. BioClip outperforms the standard clip. However, species-level separation is still challenging. This suggests that models should be trained specifically on trace data, even if they are pre-trained on large-scale animal image datasets. Here is the detection result. RT-DETR achieves the best overall results, especially for frequent categories. However, detection of rare classes is still difficult. Among trace types, feathers perform the best overall, likely due to their large category diversity. Here is the segmentation result. YOLO V8, YOLO V11, and Mask Dino perform comparably across segmentation tasks. Among all trace types, feathers consistently achieve the highest MAP, even though they include the largest number of species. This might be attributed to their distinct color appearance. Here we show detection and segmentation results on AnimalClue using YOLO V11. Green boxes indicate correct predictions, while red ones indicate incorrect ones. We observe that faint footprints or unclear traces often cause species misidentification, which reflects the challenges of working with real-world wildlife data. Here is the result for trait classification. Feathers achieved the best results overall. Interestingly, for feces, aquatic, and predator traits showed high accuracy, possibly because of clear morphological differences among species. These results suggest that trait inference depends on the trace type. Here is the summary. We present AnimalClue, a large-scale dataset for identifying animal species from their traces. It covers 5 trace types and provides over 159,000 bounding boxes from 968 species and annotations for 22 traits. We also evaluate various vision models and highlight key challenges in trace-based recognition. We hope this dataset supports future research in animal tracking and wildlife conservation.